today's lesson, we're going to focus on looking at evidence of relatedness, right? So far, we've looked at the mechanisms of evolution, how they lead to speciation, and how over many millions of years, many species have emerged through evolution and they've diverged from each other, okay? So basically, think of it like a tree, and the tree has branched out to create many branches, and each of them have diverged into their many different uh, levels, right? So basically, look at any tree outside, you see how it's spreading out. Think of each single sub branch as being one different species. So you can see how much evolution has diverged from each other, how creating a diverse group of living things. So what we are going to do today is basically looking at all of those different species, all of those different divergence, and try to find a evidence to relate them back all to their common ancestors. So remember that in the previous chapter, we said that all of them would have originated from one single cell, the first ancestor cell, and eventually that has changed into all these different life forms that we see today. So what we're going to try to do today is we're going to try to look at the mechanisms that we can use to relate them back to that common ancestor, right? Who is it that we are related to, and how is it that we can actually link those ideas of relatedness together, right? So all right, so I think we've already done this question, so I'll just skip it. Yeah, trace fossils. Can anyone tell me why it's a trace fossil? This is something we've discussed as well. Can I get, sorry, yeah, Avi? So what is the key feature about a trace fossil? Yeah. So it's indicating the presence of the organism, but it has no trace of the organism itself, right? Basically, anything like a nest, a habitat, a footprint, anything like that is a trace, because we can say that, oh, the living thing left that, the living thing built that. But the living thing itself was not found there. So that's why it's called a trace fossil. All right, so today's lesson, like I said, we're looking at the evidence of relatedness between species, and we're going to use two main mechanisms to relate them together. One is called structural morphology, and the second one is molecular homology. Okay, so the first one, structural morphology, looking at the physical features that different species have that can be related to a common ancestor. So essentially, what are features that different species, maybe even birds and humans have, that we can relate back to a common ancestor that we had many millions of years ago, right? And the second part is molecular homology. That's basically looking at the DNA makeup or the amino acid makeup of different species to see how there are what's the similarities between them, and how we can try to study that to relate it back to relatedness. So certain why this amino acid sequence study work is that we can basically find a gene that is conserved between every species or most species, and study that one gene to see how it's changed over time. And as such, we can kind of make a comparison table of how it's changed over time, and that actually indicates to us how related each species are, moving from one to the other. Okay, so these are your glossary tasks. Make sure you get that done. So when we think about relatedness, what is that word actually meaning? So when we say relatedness or how they are related to each other, we're basically trying to link them back to that common ancestor that we would have shared together, right? So most people think that we can always determine that by looking at how closely they resemble each other, right? Different species, they look alike from each other. That means they are closely related. But that isn't always going to be the case. For example, if you look at a lizard and a salamander, you would instinctively think that a lizard and a salamander are closely related because they have similar physical traits. But when you actually study their morphological makeup, you get a sense that humans are more closely related to lizards than lizards are to salamanders, because lizards they diverged at a much earlier period. Basically, once we had vertebrates, there was a divergence between amniotes and amphibians. Then from that divergence gave rise to mammals and reptiles, and then only humans and lizards. So if we actually were comparing the relatedness of humans, lizards, and salamanders, you would say that humans and lizards are more closely related, because the divergence between those two different branches happened recently compared to the divergence between the salamanders and the lizards, which happened much longer ago. So that's why we would say that humans and lizards are technically more closely related than lizards and salamanders. So that's why it is important to note that physical traits are not always the key determiner of how closely related a species are. You always have to study two forms, which is a structural morphology, where we actually study their physical traits, and the second one, which is molecular homology, how closely related are their genetic makeups, right? That is more of a key indicator than this structural morphology anyway. Okay, so those are the two primary methods that we are going to be looking at today. Let's have a look at the first one. So structural morphology, this is basically looking at the physical structures between two different species and try to establish a relatedness as a result. And there's two forms that you can take. One is homologous structure, and the next is a vestigial structure. So these two structures can be used as evidence to basically link different species as being related with each other, right? Let's look at the first one, which is a homologous structure. This is where basically the structural features can be shown to be derived from a common ancestor through similar bone structures. So this is your perfect example right here, right? The example is that with the forelimb, basically the upper limb of these different species, you can see that there is a common structure above them in terms of their bone structure, right? But they each are used for different functions. Bats use that forelimb to actually be able to fly. Whales use it to basically propel themselves forward. Cats use it to stand on all fours, and we use it for many different purposes. It's basically multi-purpose for us. But when you look at it in terms of their bone structures, you can see that there is similarities between them. But in terms of their function, look, or even size, they are all very divergent from each other. That is the result of evolution changing them. But the thing is, when you look at the bone structure itself, it hasn't changed, right? There's a humerus that is connected to the other bones that we will look at, the names we'll look at in a bit. But basically, the fact that they have similar bone structures is a key way of basically studying how they are related to each other. So these structures may look very different and may function very differently, but the bone structure is the common link. And when we try to find a common ancestor between these four different species, we go all the way back to early tetrapods, right? Primitive tetrapods. These are basically the living things that were first theorized to be able to walk on land, right? So it's said that first low fin fish, they were the ones that evolved these four limbs. Over time, we diverged from so tetrapods, they diverged into many different species, many different subspecies, and so on, to give all the way until where we are now. So in fact, if we look at it, try to look at it in a very clear way, you can see it here. So it happened around this point. So the single large limb bone articulates into two smaller bones, which is your two bones that we're looking at right now, right? This is your humerus. Need the names of them. I think it's down here. Okay, the names I can't remember right now for the bones. But essentially, that structure evolved early on in the tetrapods and has been maintained in terms of the bone structure over many different lineages, and even until today in humans and different animal species, you can find it where the bone structure, meaning that how the bones are made up, the humerus connecting to these bones, connecting to those bones, is still the same, but how they are used is very different. Their functions are very different. But the fact that the bone structure itself, essentially, think of it like alphabets, right? When it comes to chords, A, B, C, D was the formation of those alphabets, and that has not changed. The formation of those alphabets is consistent throughout. Even though how those alphabets can look has changed, or how we use them has changed, but the structure in which they are arranged stays the same. A, B, C, D, right? It's the same here as well. Okay, anyone still confused about this one?
Does that bring wood from fish? Like what?
Remember that the idea is that fish were the first living, not to say first living, but like eventually there was fish that could walk on land, and that's how terrestrial living things came to be. Life evolved in the oceans first, and then only came into land. Because think of it like this, the ocean is basically almost like a cheat code. Because when you're in the ocean, your body weight is supported by the ocean as well. So you don't have to support all the mass you have. And that allows life to emerge much more easily. Not say easily, but naturally it can happen faster compared to on land, where it needs to basically be able to fight against gravity to even emerge. And that's already a very big barrier for life to emerge. So life, in theory, there's actually a lot of evidence for it as well. Life would have emerged on the oceans first, evolved to become very divergent, and eventually there was a point where they started to become terrestrial as well. So they would have basically been able to survive in the oceans and survive in the land as well. And then over time, they started to diverge, becoming full terrestrial beings. But when you look at it, going back studying the homologous structures, you can find that there is one that can be linked all the way back to that first point, which is the fact that all of us had a forelimb, starting with the humerus, then moving on with the, or forgot the name of the bones. I'll get you guys the name of those bones in a bit at the end of the lesson. But essentially, that structure has stayed the same, even though the shape it takes has been altered over many cases of evolution and natural selection. It has changed and become some like ish, become different looking or functions very differently. The structure, meaning that the arrangement of it has not changed. All right. Anyone still confused? So yeah, basically that common ancestor is what gave rise to all the tetrapods that exist today. In fact, tetrapods, that's we are all falling under tetrapods in a way because of this classification. All right. So think about homologous structures. They function differently, look differently, and their size is very different, but their bone structure is the same. Thus, you can relate them all back to being from a common ancestor, right? Moving on, next is the vestigial structures. So these are the structures that originally served a purpose in the earlier ancestor, but due to natural selection, selecting for different features or different traits, these structures have actually lost their original function and are no longer required for survival. The best example of this in primates is tails, right? So if we look not too far, even look at monkeys, certain monkey species, they do have a tail, and that function is very basic. It's used to balance their bodies when they live in trees, right? So this is when primates primarily used to live in trees, so needed tails to balance themselves in that environment. But over time, natural selection allowed us to move on to land and allowed us to become bipedal as well, meaning that we stand up on two legs instead of all fours. And over time, the purpose of these tails became less and less important, meaning that they were no longer required for survival. As a result, natural selection basically started to shorten the tails, eventually getting to the point where all that was left of them was a vestigial structure known as the coccyx. And the question is, why do we still have that? Why even have that? Why not just go all the way and remove it? Because that is not to say a flaw, but a feature of the mechanism of natural selection. So how does natural selection work again? Let's just do a very quick recap. Basically, natural selection will always select for advantageous traits, will select against disadvantageous traits. Thing about it is that it won't select against trait that has no detriment or advantage. So basically, if it's not actually to make the living thing experience any detriment, then natural selection won't select against it. It will just persist within the living thing for many years, for many generations to come, because, like I said, right, only when there is a disadvantageous trait, something that's causing the living thing to lose fitness, it's basically its survivability and reproducibility is impacted. That is a disadvantageous trait. If it doesn't do either of those things, its fitness is exactly the same, whether it has it or doesn't have it, then there is no selection pressure acting against it. Thus, it will persist in the population and it will persist over many generations to come, right? So this can almost be thought of as an artifact of natural selection because it did shorten the tail to this point, but the fact that at this point it didn't serve as a detriment, it did not reduce the fitness of the living thing, means that it just stayed on from that point onwards. So we can actually use this as a method to infer relatedness between different species, right? So as a result of us having this coccyx, we can then relate it back to the fact that earlier primates had a tail, so we can then link it together, saying that we are our relatedness to primates is actually very close, seeing that we have the vestigial structure of a coccyx that can be related to the tail of a primate, meaning that we can basically form a link saying that we are probably descendant of early primates that have evolved to become into what we are today, right? So the fact that there is these structures that can relate them back to them is allowing us to form links between each other. And in humans, there are deep vestigial structures and organs that we can actually use to link back to other species, right? Yeah, we start off with the coccyx. Another one, male nipples, tonsils, ear muscles, the Darwin's point on the ear. I think that's, forgot which one that is. Something about your eye, your wisdom tooth. Everyone knows the wisdom tooth is a pain. In fact, it's something that is kind of selected against, but it's still a slow process. Remember that natural selection takes many, many generations, so it might get removed in many generations to come, but for now it's still persisting. It's basically a vestigial structure from our previous ancestors, appendix, and goosebumps. The goosebumps that you get, that's not really something that is serving a purpose for us. It is a vestigial trait structure that we have from our previous ancestors. Right? Any questions about this one? So simplification is that these are basically structures that had a function in an earlier species, but due to natural selection, we have lost that original function and we only have an artifact of it left within ourselves. Basically, almost like a artifact of the previous ancestors within our body that we can use to link it back to that previous ancestor. All right. Okay, moving on. So bonus information is about analogous structures. So this is not really related to what we're looking at now. It's basically something you should be able to understand as well. What analogous structures talking about is basically when different divergent species, right? Basically, they have diverged a very long time ago, millions of years ago. They somehow evolved similar traits. For example, when we think about a moth, which is an insect that diverged from the main branch a long time ago, and then we have something like a bird or a bat. These are mammals, right? Birds, and then we have bats, and bats are mammals, and yet they all have the trait of being able to fly. Why? It's because that over it's basically an evidence of convergent evolution. Basically, they have independently evolved similar traits to adapt to similar environments and selection pressures. Basically, there were selection pressures that were acting on them in a similar fashion, selecting for traits that allow them to evolve flight. But the fact is, they did not have any common ancestry for a very long time. So this just happened due to the fact that they had similar selection pressures that were selecting for similar traits, and they all developed a similar trait to be able to fly. So even a key way to determine whether it's analogous structures or whether they were related is basically look at how different the structures are. They are going to look very different between each other. For example, with a moth, they use a basically a one structure wing that is very lightweight, whereas a pterodactyl uses almost like a skin layer to flap the air. Birds use feathers, and bats use something similar to pterodactyls. In fact, if you try to look at it, pterodactyls and bats, they have that similar way of using a flap of skin to be able to fly. But the fact is, all of them have the same trait, which is being able to fly. But all of them evolve it separately, unrelated to each other, due to similar selection pressures, just giving them that selection advantage. Okay, and again, this isn't really important to know to this topic, but it is something you should know in general because it will come back later on. Basically, the word convergent evolution will be necessary for you to know, and this is something that is important to that. Okay, so the next one. So far we've looked at structural morphology and how we can use that to link back to relatedness, right? Both in terms of their vestigial structures and also homologous structures. Now we're going to study their genetic makeup. And this is a much more important concept in modern evolution because this is basically much more testable and much more provable in terms of looking at how they've changed over time. So this is basically focusing on the difference of DNA and amino acid sequences to be able to relate each other to see how related they are. And this is actually much more easier to do than you might think. So if I showed you purely like this and don't give you any other information, right? So we have so we have 4 different amino acid sequence here for the same protein. So this is all going to make hemoglobin, but their amino acid sequences are slightly different due to mutations and so on, creating those slight differences. So if I ask you, do you think humans and gorillas are more closely related than humans and chimpanzees? What would your answer be? Purely based on what you can see here. Okay, can I get Janita? Which one do you think are more closely related? Human and chimpanzee or human and gorilla? So which ones? Are they all the same? Are you sure? Look at the gorilla's amino acid sequence, look at the human, and then look at chimpanzee. So human and chimpanzee, you think are more closely related. That's it. It's as simple as that. Basically, look at how many differences there are in the amino acid sequence. And as a result, you can basically determine how closely related they are as well. Right? So basically, when you study this, you can definitely already see that humans and chimpanzees almost have the identical, basically have an identical amino acid sequence to make hemoglobin. That means they are much closely related when studying using a amino acid sequence. And then as such, we can also say that humans and gorillas are less related compared to chimpanzees, because you can see that there is differences here and here between the humans and humans and gorillas. So that means that there is a further relatedness compared to chimpanzees for humans. So when you start to talk about relatedness, you're always going to be comparing it relative to one and another. So all you have to see is between this and the one you're comparing it against, how many differences there are. If there are no differences, they are very closely related. And doing the same thing between humans and gorillas, how many differences there are? There's one. That means they're slightly less related compared to chimpanzees. The number of differences determines how further related they are. So the more differences you have, the further related they are. Okay, the lesser amount of differences you have, the more closely related you are. Okay, so essentially, why is it we can use amino acid sequences? Is because there are basically some genes known as conserved genes, right? These are genes that have remained largely unchanged between different species, and they can actually be found in the genome of many different species, right? An example of one is hemoglobin. So you guys have probably heard of hemoglobin. It's a protein within your body that allows you to basically carry oxygen all from your lungs to all your cells. So it can be found implanted within your red blood cells, and without this, you have a lot of issues and a lot of problems. Basically, without this one protein, you won't be able to get oxygen to all your cells adequately, and it can actually cause a lot of issues. In fact, there is a very common disease associated with not being able to produce hemoglobin properly. Cystic fibrosis, I think. Was it? Oh, wait. Okay, I'll come back to that in a bit. Basically, they even found a gene therapy using CRISPR to allow you to produce hemoglobin properly, because the disease that it's curing is caused due to the fact that they can't produce hemoglobin properly. So it's causing them to not be able to get oxygen to the cells adequately. So they came up with a recent gene therapy to allow them to produce hemoglobin in a proper form that allows them to actually fix that issue, right? Fix that genetic disorder. So even if the gene is largely conserved between different species, certain amino acids can be altered to still give you the same protein, right? And by looking at how those changes have occurred and how many changes have occurred, we can basically link the relatedness between the different species. And this isn't only for hemoglobin. We also have another example, which is your cytochrome C, which is an enzyme present in mitochondria that has a key function in the mitochondrial DNA. So same thing, even if it is largely conserved, the changes can be used to study the relatedness. So the more differences they are, the further relatedness. Okay, I'm going to go it the other way. The lesser the differences they have between each species, the more closely related they are. Okay, so in fact, we have a point where we have studied it here already, and as a result, we already know which species are closely related to humans, right? By comparing these different amino acid sequences for the same conserved gene, you can see that human and chimpanzees, there's no differences. So we are most closely related to chimpanzees. Between rhesus monkeys, there's one difference. So the next closely related to us is rhesus monkeys, so on. And you can see that you can go all the way to wheat even and form a link saying that we do share some relatedness to wheat, because even in wheat, there is that conserved gene of being able to produce cytochrome C, but there is a lot of differences. So that shows that it diverged super early on to become very different between each other. Okay. So basically, the more the differences you see, the further the divergence would have happened. For example, think of it like the tree branches, right? If this is your main stem, when the divergence happens, you have two different branches forming, right? So think of this as basically where wheat and the primate where we originated from happen, and this is why the differences between that one gene has accrued so much, because the divergence happened much early on compared to something like for rhesus monkey and humans. The divergence would have happened much more recently, basically a few million years ago only, because we've only accrued basically one difference, and between chimpanzees and humans it would have happened super recently. Okay, if this is a few 100 million years ago, this would have been actually a few million years ago. Of course, don't quote me on these dates. These are just estimations based on how it could look like. But the fact is, you can think of it as being able to form a kind of a puzzle showing that when they would have diverged from between each other from their common ancestor. Okay, we would have had a common ancestor that allowed basically wheat formed up here, but wheat and us shared a common ancestor at this point, which diverged to give different lineages, and over time wheat formed here and we formed here. So that's why we can link it back to that common ancestor by studying the amino acids and saying that it diverged a very long time ago. So our common ancestor would have been many, many hundreds, if not billions of years ago. All right? Anyone still on the fence about this? Okay. So if we can do that with amino acid sequences, okay, let me give you an idea of the limitation of amino acid sequence, right? So far, like I said, we can use the differences between the amino acids to study relatedness. Yes, we can. But if we look at this right now, between chimpanzees and humans, it says there are no differences. But obviously there are differences, right? In fact, if you study the genome of a human versus a chimpanzee, almost 90+% of the DNA between us is going to be similar. But that remaining percentage is what drives us to become so different between the two species, chimpanzees and Homo sapiens. They're super different because of that final percentage, and it won't always be able to be studied using amino acid sequences because of how closely related we are. So the next best thing to use is the DNA sequences. Okay, so this goes on another level of specificity because DNA sequences are what make up amino acids. Every codon codes for one amino acid. Remember, 3 by 3 by 3, that's how we read it. Each three nucleotides are going to form a codon, and one codon codes for one amino acid. Hope everyone still remembers their genes, like the basics at least. So this is going to come back to that, right? Basically, looking at this, DNA sequence of different species can be compared to determine the evolutionary relatedness between each other. So this is going to be much more detailed compared to amino acids sequence studying, for amino acid sequence relatedness, because between different species, even if they're very closely related to each other, they will still have a lot of differences in their DNA sequence. Their amino acid sequences might look very similar, but in the end, their DNA sequences will show a lot of divergence from each other, right? So when we look at this, the higher the similarity of DNA sequences, the closer the level of relatedness we can say between those different organisms. We can compare the differences in the DNA sequence of different species by simply looking at the nucleotides that make up the DNA, right? Remember the 4 nucleotides? What are they? Adenine, guanine, And last one, put right. Yeah, those are there, of course, but yeah, 4 nucleotides, and they make up your genetic code. And by looking at that genetic code of different species and comparing them between each other, we can also study the relatedness of different species, right? So if we look at this one right here, right? Yes, can't really show the whole thing. So okay, this one's good enough. All right, if I asked, let's see, everyone try to tell me humans, which one are they most closely related to? Rats or yeast? Everyone had, I'll give you 2 to 3 minutes to think about this. What? So, which species, rats or yeast, are humans most closely related to based on DNA similarity, DNA sequence similarity? Okay, so look at the two different three sequences, see how similar they are. As a result, try to find out which species are more closely related to each other. Does everyone have an answer? Are you sure? Well, I don't know. You should know what you're doing. All right, let's break it down into the simple steps, okay? Between humans and rats, how many differences are there? 3, right? Basically, one here, and one here. So 3 differences you can see. Between humans and yeast, how many differences can you study? 1, 2, 3, 4, 5, 6, 7. So which one has more differences? The yeast compared to humans. So as a result, we can make a conclusion that rats are more closely related to humans than yeast. Right? Basically, count the number of differences. The lesser differences means that they're more closely related to each other. It's that simple for this thing. Okay. So like I said, why would we use DNA sequence studying compared to studying amino acid sequence? Is when we are studying closely related species, amino acid sequences can be misleading. So due to the high level of conservation within amino acid sequences between closely related species, sometimes we can't compare the different the same amino acid between different species. So instead, this is where we compare the nucleotide sequences and see, look for even the slightest distinction between those species, because sometimes silent mutations do occur where the mutation happens and the amino acid, the nucleotide is changed with another nucleotide, but it doesn't cause any change in the amino acid production, right? Basically, the same amino acid is produced, but in the sequence itself, the nucleotide has been changed. So only by studying the nucleotide sequence could we have seen that difference. We wouldn't have noticed it if we only looked at the amino acid sequence. Does everyone understand what I mean? Okay, let me try to indicate this in the best way I can. I have a picture of it actually. Nope. I will put it up later. So basically, so think of it like this, right? Let's say the code, let's say the code was A, C, G, T, C, G, C, A. And this codes for methionine, lysine. I need an amino acid. Does anyone know any amino acids? Okay, so okay, I'll just put another lysine for the sake of it. Actually, I'm just gonna write a random letter to say that's an amino acid. Better that way. Yeah, I'm pretty sure that's an amino acid. Okay, So these three codons code for these three amino acids. Okay. So now a silent mutation has occurred where A, C, and now the G has changed into a C, but everything else is the same. G, C, A. Okay, this is referred to as a point mutation. Specifically, when we look at it, it's actually going to be a silent mutation. Why is it considered a silent mutation? Can anyone tell me right now? Looking at this and that, why is this referred to as a silent point mutation? So that's a point mutation, but why is it considered as silent? Exactly, right? The amino acid that was produced as a result of that codon is still the exact same amino acid that was originally produced. So if we only studied the amino acid sequences, we would say that they are very closely related. There are no differences at all. Only by studying the nucleotide sequence, we can see that actually there is one difference, right? So that means that there is a one difference, meaning that they are very closely related, but they're not exactly the same, right? So that's why nucleotide sequence, DNA sequence studying can be much more detailed compared to amino acid studies, because between closely related species, there can be those point mutations that are silent that cause the amino acids to be exactly the same. The only time Sorry, not the only time. The time that amino acid sequences are very useful is when we already know for a fact that the two different species are very not closely related to each other. That way, amino acid sequence studies can be a quicker way to determine how closely related they are. But when we're dealing with very closely related species, the best thing to do is to compare the nucleotide sequence. I hope everyone has a good understanding of this, or a decent understanding. We'll go. So, yeah, like I said, amino acid sequences, however, are easier to interpret and are therefore used to determine relatedness between more distantly related species, because they are basically much more easier to decipher. Because when you're trying to sequence a genome, you have errors that can occur and uncertainties that can occur as well. So amino acid sequences, to interpret that, you get a better chance to do it. So it's better to use between not closely related species. So other than DNA and amino acid sequences, we can also instinctively just compare the entire genome of a living thing, right? The entirety of your DNA, we compare it with another species to see how conserved or how different it is. So essentially, it's the same principle. The higher the degree of similarity between the genomes of different species, the more related they are, and the more recently they would have diverged, meaning that their common ancestor would have been much more recent, and that when they diverge into giving that two different species. So, oh yeah. So remember how I kept saying chimpanzees and humans are very closely related? So when we're looking at the percent of amino acid that are identical to the amino acid in human hemoglobin peptide. So when we're studying the amino acid sequence of hemoglobin, we can see that there is a very high relatedness between a chimpanzee and a human, meaning that that one amino acid sequence is highly conserved between these two species, meaning that it's very not indicative of how closely related, not indicative of their differences. So we end up needing to compare using something like DNA sequences or the entire genome sequence to actually see how further they are in terms of relatedness. Because this is not giving us a lot of data. In fact, it's saying that these two species are very, very closely related to each other. So to actually get useful data, we need to go deeper into it and actually look at the DNA sequence, the entire genome sequences to see the differences, and then we can tell how closely related they are and when they would have possibly diverged from each other. But then something like frog and humans, we can see that there is a low level of conservation for the same amino acid sequence. So we can actually use the fact that that is the case to compare relatedness between a frog and a human versus other species, where humans versus chicken and human versus frog, we can see that human versus chicken have a more conserved amino acid sequence. That means they are more closely related to the humans. So they would have diverged much more recently compared to a frog. Okay. Remember, the further they are in terms of relatedness, the longer it has been since we diverged into becoming different branches. Okay. So think of it in terms of the trees again, right? So the trees that branched out at the bottom compared to the branches that branched out at the very top. If you were going to think of it in terms of relatedness, you would say that the branches at the bottom have very little relatedness to the branches on top because they diverged between each other much earlier. But comparing to the branches on top, the ones that branched out at the very top, they're much more closely related to each other because they only branched out much more recently, right? Okay, hopefully that makes sense. All right. So by studying these, basically by studying how closely related they are, we can start to do something which is building a phylogenetic tree. The whole point of analyzing, determining relatedness is so that we can construct a phylogenetic tree and determine the evolutionary relationships between different species, right? Based on certain information. So let's say we are using cytochrome C and we are determining what are the differences that they have, right? So when we do such a thing, we can basically say that we know that rats are more closely related to humans and that yeast was the most distant related to humans. So we know that our common ancestor with yeast would have diverged much earlier compared to rats, which would have diverged much recently as a result of us being more closely related to them. Okay, simple, always associate this idea together. The more closely related we are, the more recent the divergence happened. The further related we are between a species, the longer it has been since we diverged for them, between them. Okay, so we need to use that principle to be able to construct phylogenetic trees later on. Like I said, right? Building a phylogenetic tree. That's why this is all very useful. So far, in summary, the two things that we looked at, structural morphology, molecular morphology. For structural morphology, there are two main things, homologous structures and vestigial structures. Homologous is where they, even though they are different function between different species, they still have a similar bone structure, or a similar underlying structure that can be used to study relatedness. Next is the vestigial structure, which is basically a remnant of a previous species that is still remaining as a result of natural selection, not removing it, right? Basically, they remain within a species despite losing their function and necessity, and they can also be used to study a common ancestor. That is for structural morphology. For molecular homology, we study amino acid sequences and DNA sequences. For amino acid sequences, it is not as accurate if we're dealing with a very closely related species, because between closely related species, they can be conserved in terms of their amino acid sequences. So we would go further detail with a DNA sequence study, which shows even any point mutations that could even be silent, that couldn't be detected when you do a amino acid sequence study. The only reason we would choose an amino acid sequence study over a DNA sequence study is because the amino acid sequence study is much more easier to interpret and also easier to basically do